Greetings! In this module, you will learn how to use the web app Climpart, a tool to support seed collection for conservation efforts. My name is Kyle Doherty, ecologist, software developer, and author of the app. In this module, we will learn the underlying principles of climate similarity, review app features, conduct an example analysis for a region of interest, and discuss basic and advanced applications of this approach. Conservationists often wish to prioritize genetic diversity when collecting seeds and other plant materials. Climate is a selective pressure in plants and can be a driver of genetic differentiation leading to local adaptation. Thus, an approach to capture genetic diversity is to collect from a breadth of climate conditions. One tool to guide collectors to areas of unique climate is the app Climpart, discussed in this learning module. This tool's primary application is for seed banking efforts, though it may also be useful to research ecologists studying the effects of climate gradients on ecological phenomena. To understand how Climpart operates, we must first understand the concepts of climate space and climate similarity. We will examine the state of Utah and the United States as an example. Here we extract two pieces of climate information, mean annual temperature and precipitation for this region, and plot them in the panel on the left. We refer to the resulting cloud of points as the climate space. Each point in climate space has a corresponding geographic location, a 1 kilometer by 1 kilometer pixel in Utah. Let's pick a point in southern Utah, the white dot in the panel on the right, and compare its climate to that of areas throughout the region. We can plot this point in climate space and see that it receives 300 millimeters of rainfall and is 7 degrees C on average. Regions on the map most similar in climate to this point, those adjacent in climate space, are depicted in red. Note that even though a region may be geographically distant, those regions may still have high climate similarity to our point of interest. The most dissimilar regions are depicted as blue. In the context of plant materials, we might observe that a collection transferred from the origin of the white dot to blue regions on the map performs poorly relative to its home conditions. We can map climate similarity to multiple sources. Here we have performed this exercise for 3 sources, a Session 1, Session 2, and a Session 3, depicted as purple, green, and red points. Regions are colored depending on whether the climate is most similar to a Session 1, Session 2, or a Session 3, in a process we refer to as assignment. Notice also that there is a color gradient within each assignment, various shades of purple, green, and red. This gradient depicts the range of climate similarity within an assignment, where darker, more saturated colors indicate greater similarity to the point to which it is assigned. Unsaturated regions indicate transition areas from one assignment to another. In the context of plant materials, each point may represent the provenance of an available seed source. In the assignment map, a suggested seed source for revegetation efforts throughout the region. We can also rank seed sources by their similarity to a region of interest, and use this information to select multiple sources rather than simply the closest match. Recall that we may capture genetic diversity by collecting from a diverse set of climatic conditions, but how do we decide on what conditions are representative for a region of interest? The Climpart app uses an algorithmic process to answer this question. Practitioners first decide on a target number of collections and a region of interest, which they provide to Climpart. The app then identifies a set of points in climate space and geographically that best represent the region of interest, referred to as climate centers. Climpart then maps out their assignments to guide collection efforts. For more details on this method, please refer to Doherty et al. 2017. Full citation found in the references at the end of this module. In our example, we looked at 2 aspects of climate, mean annual temperature and precipitation. However, climate similarity analyses may be conducted on any number of climate variables. Climpart conducts analyses on 7 climate variables that each contain unique and potentially ecologically relevant information on the North American continent. The selection process for these 7 variables is described in Doherty et al. 2017. These variables are a subset of the BioClim dataset, including mean annual temperature, diurnal range, temperature seasonality, temperature of wettest quarter, mean annual precipitation, precipitation seasonality, and precipitation of warmest quarter. For more information on how these variables are derived, visit the address above or refer to Hidgmans et al., 2005. The address to the Climpart app is linked above. When you navigate to Climpart, you get a welcome message stating that the app is initialized, a link to the underlying paper upon which the analyses are based, as well as instructions on how to run the app locally on your computer. We will get to this in a moment. However, the online version is limited in computational resources, so very large spatial analyses may not be appropriate for the online version. After clicking through the welcome message, you will see the graphical user interface of the app. In the center of the app, you will find an interactive map. To the left, you have controls to execute your analysis. And at the top of the screen are a few tabs for additional analysis outputs, a brief background on the app, and the instructions for running the app offline. Let's take a look at the controls working from the top down. The first items ask you to define your area of interest, the region in which you will conduct your analysis. At present, the extent is limited to North America, excluding larger bodies of water and a few islands. The default setting is to select your area of interest with two sliders that define the upper and lower bounds of latitude and longitude. If you click and drag an endpoint on a slider, you will notice that the extent of the green box on the interactive map changes to reflect the limits you select. You may also define your area of interest by uploading a spatial polygon. This could be a species range, ecoregion, jurisdictional boundary, or any map unit of interest in North America. You must provide a file compressed in the .zip format, which contains 4 components listed above of an Esri shapefile. Search for .zip file compression if you are unfamiliar with the process. The app accepts spatial polygons of any coordinate reference system. After a few moments, a polygon will render on the interactive map depicting the bounds of the spatial data you upload. Here we have uploaded a polygon of the Great Basin ecoregion, which we will use for an example analysis. We will leave the other settings as is for the moment and begin our analysis by clicking the green Generate Partitions button in the Controls bar. You will notice progress bar messages appear in the lower right-hand corner of the app, which indicate that the analysis is underway and which give an indication of the underlying methods as they are implemented. Analyses for ecoregions of this size usually complete within a minute. However, this is somewhat dependent on the level of traffic the app receives. Analyses of larger spatial extents, or those with a greater number of partitions, will take longer. The servers hosting the app may run out of memory and crash if the extent is very large. If you find that your analyses continue to crash after multiple attempts, this is an indication that you'll need to run them offline on a computer with greater resources. More on this in a moment. If you receive this message early on, you have selected a region for which the app has no climate data and cannot conduct an analysis. When analyses are complete, the map will center on the spatial extent of your output. Here we have identified 5 climate centers in the Great Basin ecoregion. These are represented as colored points on the map, with the climate center ID represented in the legend. You may mouse over the climate centers to reveal their ID numbers. While climate centers differ in their climatic properties, their ID numbers have no significance other than to assist with organization. During the analysis, the areas within the Great Basin ecoregion were assigned to one of the 5 climate centers that most closely resemble the climate properties of that center. These assignments are depicted as a colored overlay. The coloration of an assignment and its ID matches its corresponding center, and you may refer to the legend key to discern among assignments. If at any time we wish to turn off the overlay to reveal the underlying base map, we can do so by clicking the Overlays checkbox in the upper right. If we zoom in on Center 1, we can see the similarity gradient within areas assigned to it. Darker shades of burgundy indicate higher similarity to Center 1. Lighter shades indicate lower similarity. If we wish to examine the conditions at any point on the map or confirm assignment and similarity values, we click on the map and a pop-up appears. The Climate Similarity Legend key indicates the range of similarity values for your analysis. A similarity value of 100 indicates identical climate conditions to the center, and lower values indicate differing conditions. A value near 0 would indicate the lowest similarity possible within the region of your analysis. It is often the case that a climate center is difficult to access or that there are no target plants at its location. Here the utility of the Climate Similarity Index comes into play, as the darkest regions of an assignment will have nearly identical conditions to a climate center. Thus, the most effective strategy for seed collection or sampling efforts is to target the darker regions of an assignment, rather than the centers, to ensure that we capture the climate conditions of interest. If we click on the Climate Center Data tab at the top of the app, we can get additional information about the results of our partitioning efforts. This tab contains coordinates and climate properties of the centers produced during the analyses. This is one way we may assess our results. In the context of climate centers, partitioning determines the separation of climate properties between climate centers. Let's investigate the partitioning concept in detail. Looking at the mean annual temperature column, we see that the climate centers range between 6.5 and 9.9 degrees C. Notice that centers 1 and 3 only differ by 0.3 degrees. However, when we look at the mean annual precipitation values, we see that these two centers differ by 91 millimeters, a substantial amount for this cool desert region. This illustrates the inner workings of Climpart, which partitions 7 climate variables by default. Centers that share similar values for one or more properties will differ in another property. Note that you can sort these values to help visualize the degree of separation from one center to another along a climate gradient of interest. Now let's click on the Within Assignment Distributions tab at the top of the app. After a loading period, you will see 3 box and whisker plots depicting the climatic distributions for our 5 assignments. This is another way in which we can visualize the granularity of the partitioning effort. In the context of assignments, partitioning determines the degree of overlap in conditions between climate center assignments as well as the range of conditions within assignments. If we look at the top panel for mean annual temperature, we see that the median value for Center 2 assignment, the green regions on the map, is about 10 C, and that the bulk of the data lie 0.5 C above or below this value, with a few outliers ranging a few degrees or more. You can inspect the distributions of any 3 climate variables simultaneously, switching between them in the drop-down menus at the top of the window. Wide-ranging values within an assignment, or overlap between conditions of 2 or more assignments, may indicate we may want to amend our analyses. If we decide that we would like a greater degree of separation between conditions of climate centers, less overlap of conditions between assignments, or a tighter distribution of climatic conditions within an assignment, we can increase the partition number. After setting the partition number, click on the Generate Partitions button to rerun the analysis. Here are the map products. In general, the geographic extent of assignments will decrease as we increase partition number. After rerunning the analysis for 15 and 30 partitions, let's visualize the results in the Climate Center Data tab, focusing on mean annual temperature data of our centers, which we have sorted from low to high values. In our initial 5 partition analysis, we see that our center values range from 6.5 to 9.9 C. At 15 partitions, the range increases from 3.3 to 10.6 C. Finally, we see a range of 2.9 to 12 C with 30 partitions. So as we increase the number of partitions, we better capture the extreme ranges of the gradient. Additionally, steps along the mean annual temperature gradient become smaller and more numerous. Note that climate centers are not shared as we increase or decrease the number of partitions. Each has its own solution, its own set of climate centers. We can also increase the partitioning granularity of a subset of the variables by giving them greater weight in the analyses. We adjust weights by clicking the Weight Variables button in the controls. When you click the Weight Variables button, the controls will expand with 7 additional sliders, each dictating the weight of the corresponding variables. Here we set all weights to 0 except for mean annual temperature and precipitation. Setting variable weights to 0 excludes them from the analyses. By assigning weights of 0 to all but mean annual temperature and precipitation, we can tighten the distributions of those focal variables in each assignment and lessen the overlap across assignments. Note that the distributions of the third variable, precipitation seasonality in the weighted example, are both relatively wide and overlap more with one another. If we had instead decided to keep information from the other 5 variables, but reduce their importance rather than exclude them entirely, We might set their values somewhere between 0 and 1. Setting variables to 0.5 would equate to half the importance of an unweighted variable, 0.25 a quarter, and 0.1 a tenth. When is it appropriate to weight variables? You might be conducting a natural survey studying only a subset of the variables, or you may have prior knowledge of a species' response to climate transfer, derived from a common garden or reciprocal transplant study, and wish to establish transfer guidelines for that species for a target number of plant materials. If you possess empirical data such as this, you might first model plant performance as a function of climate transfer across the 7 Climpart variables, using an algorithm such as random forest, maxent, or boosted regression trees. The next step would be to extract the variable importance metrics from the model, then identify the maximum importance value, and finally divide all importance values by the maximum. These relativized importance metrics could then be provided as input weightings to Climpart. After running an analysis, you can download the relevant outputs to your computer to explore in your own mapping software. To reproduce the aesthetics in the interactive map, set the SimVal TIFF to a black to white gradient, then overlay the center assignment TIFF, setting it to 50% transparency or as desired. Center coordinates and climate information are found in the center data CSV. Box plot figures for the assignment distributions may also be found in the PDF file. We now provide the option to run Climpart on your own computer for larger analyses. Instructions for this process may be found in the Run Offline tab. Installation requires some knowledge of the R programming language, in addition to a few actions in the command line terminal. We will update this tab in the future if we can develop an alternative, more accessible solution. If you are a programmer or scientist who would like to explore the source code of the app, it is available in the GitHub repository linked in this tab. Please note that while you can leverage the power of your personal computer to conduct these analyses, you still require an internet connection to display the base maps. There are numerous potential directions we might take this app, one of which is to incorporate future climate data, which could be used to assess the future utility of accessions and plan for assisted migrations. There are also now globally available soil grid data, which could be added to study the importance of soil chemical and physical properties. It would also be beneficial to extend the coverage of the analyses globally and make them quicker. If users encounter any bugs, have questions, or wish to collaborate in future efforts, please click the Contact Info button in the app and send an email to the address provided. Climpart is a tool that provides spatially explicit guidance for seed collection. We also serve a complementary app, Seed Selector, linked above, which is primarily a tool for picking among available sessions for use in restoration projects. I'd like to thank the U.S. Geological Survey and Colorado Plateau Native Plant Program for supporting the research and development that went into these products. I'd also like to thank the Botanic Gardens Conservation International for assistance with developing this learning module.